is I want to tell you about an experiment that was done back in the 1700s. Maybe it's an experiment you would want to create at home. Perhaps you already recreated this experiment at home in your bedroom, in the corner. Your parents yelled at you. So let's just read a little bit about this experiment. This is the actual writing of Jean-Baptiste Van Helmont. He said, if you press a piece of underwear soiled with sweat together with some wheat, and you place it in an open jar, after about 21 days, the odor changes. And the ferment coming out of the underwear, penetrating into the husks of wheat, turns into mice.
Awesome.
But what is more remarkable is that mice of both sexes emerge. And these mice successfully reproduce with mice born naturally from parents. But what's even more remarkable is that the mice which come out of the wheat are not small mice, not even miniature adult or aborted mice, but adult mice emerge. So Van Helmont did this experiment. He put a bunch of his sweaty underwear and some husks of wheat in an open-mouth glass jar, left it in the corner of his bedroom for 21 days. He did notice the odor change, and coming out of this sweaty underwear wheat pile were adult mice. So,
I feel like he was just lazy.
Can you get like a good explanation?
That name just sounds
so if you recreate this experiment in your home, okay, would you see mice emerge?
No.
Maybe.
I mean, probably
Did the sweat smell from the underwear
Yeah.
turn the wheat into mice?
100%. Probably not.
Probably not.
Yeah.
Definitely not.
This is a
So, what was, yes, and that may seem kind of silly to us, but that is what he did, that is what he observed. So, what do you think was really going on here? So, if you don't think that the ferment of stench from underwear turned the wheat into mice, what really probably happened? Drew?
The smell attracted the mice into it.
Okay, and let's go a little further. Ali?
The open mouth jar left an open pocket for the mussels to sneak in.
Okay, so yeah, probably adult mice were attracted to the odor of wheat and sweaty underwear together. I don't know which one they're attracted to more. But they probably crawled into that jar. He couldn't watch it, sit there and stare at the jar for 24 hours a day for 21 days. So probably mice climbed in, were attracted to the smell, and that's what he saw coming out, was that mice had emerged from it. So what was probably wrong with how he set up this experiment? How could he fix this? Jake?
Close the lid.
Yeah, if he just had a cover on it. If he had covered the top of this jar, would he see any mice emerge?
No.
Oh. All right, so this probably seems a little silly, but this concept that non-living material can turn into living things was actually very common. Most people believed that it was possible for non-living matter to turn into living matter. So why would people, we probably think it's silly, but why would people believe this? Well, it did agree with certain things that they saw. Van Helmont did see the mice coming out of the sweaty underwear jar. Okay? And he believed in this concept that we call a spontaneous generation. The concept that living organisms could arise, could come from, could be created by non-living matter. It's called spontaneous generation. So one of the things that people saw that led them to think this is maggots appearing on meat. If you take a piece of meat and you just leave it outside somewhere, after a couple days, what you will notice is that these worm-like creatures just sort of appear on the meat. People thought that given those conditions, the meat turns into maggots somehow. There were no maggots there before. They couldn't see any maggots on the meat. They never saw maggots crawl into the meat, but somehow a couple days later, there's maggots present. So the meat must be turning into maggots. They noticed that if you have bread that's left out, eventually, after a few days, it starts to develop this living organism on it. This hairy green and blue stuff that grows and spreads from piece to piece. They didn't see any of that, anything like that moving into or crawling onto the bread. It just happens to form. So they thought the bread turns into this mold creature. If they had some soup and it was left out uncovered, it would start to become cloudy. It would start to smell bad. It would make you sick if you drank it. It was developing into this living organism, these living things that we call pathogens or germs. In the springtime, around ponds and streams, frogs would just emerge from the mud where there were no frogs before. So people thought that the mud could turn into frogs in the springtime. These are maggots. Has anyone ever seen maggots?
Yes.
Yeah. I've seen them lots of times, or several times, in our garbage can outside. Like if you throw, so we have garbage outside where we pick your trash bags and stuff. But every once in a while, if we throw like a container that had meat in it, sometimes I'll go to open up the lid and there's maggots crawling all inside of it. It's pretty gross. Mold on bread. Really a ton of mold on bread. Spoiled broth. Frogs emerging from the mud. So, are these statements correct? Does meat turn into maggots?
No.
Does bread turn into mold?
No.
Does soup turn into bacteria, microbes?
Yes.
Does mud turn into frogs?
No.
Could you come up with an alternative explanation for many of these things?
Yes.
I'm not going to ask you for it yet. Because we're going to talk about some of the scientists that actually did experiments, scientific experiments, to show that some of these things were false, that spontaneous generation does not happen. Okay, so we'll talk about 3 scientists today. The first one is Francesco Redi, an Italian scientist working in the middle of the 17th century. He was one of the first to study spontaneous generation in a scientific way. And so what we're going to do for each of these scientists is kind of go through the scientific method, our steps that we talked about a couple months ago. So, if we think about an experiment, we start with a problem. What do you do in the problem stage of an experiment? You ask a question. The question Francesco Redi was asking was, where do these maggots come from? Now, many people believe they came from the meat, turning into them. Redi had a different hypothesis. Redi's hypothesis was that the maggots actually are coming from flies. So he did an experiment. He took a few jars, and what he did is he put a chunk of just raw meat in there. One of the jars was just left open. One of the jars was covered in gauze, which let air go through, but the flies couldn't go through. And the other one was sealed up completely. And then he observed these after several days. What he noticed is that there were flies around these jars. And in the open jar, flies would go into the jar and land on the meat. And he observed them laying their eggs on it. Later on, maggots appeared. jar. Flies were seen nearby. However, they couldn't get into the jar because it was covered in gauze. But they were attracted to the smell. They landed on the gauze. And then maggots appeared there on the gauze. In the sealed jar, no maggots developed anywhere. So what conclusion do you think Francesco Redi made based on this experiment? Guys? Yeah, his hypothesis was correct. Maggots do come from flies. Maggots are fly larvae. Many insects go through these stages, a larval stage and an adult stage. Just like a caterpillar, what is an adult caterpillar? Butterfly. The caterpillar is the larva stage. The butterfly is the adult stage. For flies, a maggot is the larva stage, which hatches from the egg. The fly is the adult stage. So next time you see maggots and flies, don't think of them as gross, think about it as butterflies. Little tiny butterfly-like creatures. Sorry. And so flies find dead, rotting material, because maggots consume, when they lay their eggs on rotting meat, rotting flesh, the maggots that hatch from the eggs are very, very small, but they consume the decaying tissue and grow larger until they're ready to metamorphose into flies. So often, flies, they lay eggs in roadkill. You'll see, like on a dead squirrel on the road, if you look carefully at it, you'll see maggots crawling around there, consuming the dead and decaying tissue. Eventually they turn into flies. Flies are attracted to the smell of rotting meat. Some flowers that are pollinated by flies even produce a disgusting odor of rotting flesh to attract flies. Sort of just like a caterpillar, where it's a briefer stage. It's where they basically change from that maggot form into the adult. So Francesco Redi helped us to understand meat does not turn into maggots. The maggots actually come from flies, other living organisms. Okay, why do I have this place? Dark hearing, messed up my notes. Okay. The next was another Italian scientist working about 100 years later, Lazzaro Spallanzani. Lazzaro Spallanzani was a scientist who was studying what they call broth, basically like soup. And like I said earlier, people noticed that if you took some broth and just left it out, it would turn into these microbes that started growing, living organisms that would smell bad, it would make you sick if you ate it. So people thought it was that the broth was turning into these microbes. Spallanzani studied that question. He wanted to know what caused these microbes to form in this broth. His hypothesis was that it was not the broth turning into the microbes, but these microbes actually are coming into the broth through the air and then growing in the broth. So he did an experiment, of course. In his experiment, he took some broth and he heated it up. Then he turned off the heat, let it cool. One flask he left open to the air, and the other flask he sealed. What he saw was that in the open flask, after waiting a little while, microbes started growing. However, in the broth that was heated and then sealed, he left it for the same amount of time and no microbes grew. However, if he then opened the flask, allowing air in, letting it wait a little while longer after that, the microbes did start to form. So what do you think his conclusion was? Ali?
Microbial life comes from air.
Yeah, that microbial life comes from the air. So why didn't this one develop any microbes? Why didn't they grow in the bottom class? Tyler?
So it's sealed shut, so nothing's inside.
Yeah, and in the initial section, the high temperature killed any microbes that were already there, and then sealing it prevented any new microbes from getting in. So the last scientist we'll talk about is one you may have heard of before. He was not Italian, even though the other two were Italian. He was, you want to guess? Ella?
French.
He was French. Louis Pasteur was doing his research in the middle of the 19th century. And he, again, was studying a similar problem as Spallanzani was studying. However, his hypothesis was different. Pasteur was also asking, where does this microbial life come from? But his hypothesis was that microbes were actually not floating around on their own in the air. Do you know what he thought? Oh, I guess I'll give it to you. He thought
that microbes were carried by dust and not air.
So he thought the microbes were actually kind of hitching a ride on dust particles that are floating in the air. So that's a pretty tough one, you might think, to prove, though, to study. How could he study if the microbes are in the air or on dust particles? Well, he did this in a pretty smart way. What he did is he made some special flasks that had, it was called a swan's neck flask, because it had this S-shaped curve of glass at the edge. So he started again with some broth in a flask, and he boiled it. Now this swan's neck flask was open here, so air could still get into the broth. However, dust out of the air could not make it through the curve. All of the dust settled here on that first curve. So air could get in, but not dust. And so he heated the broth, allowed it to cool, and let it sit there. He let it stay for years, and no microbes grew. Even though air was getting in, microbes were not growing there. Then he tipped the flask so that some of the broth mixed with some of the dust and tilted it back. And then within a few hours or days, microbes started growing there. So that shows that he was correct, that the microbes are hitching a ride on dust particles. They're not just floating freely in the air. And so we know today this concept of spontaneous generation is false. Living things do not come from non-living things. All of the examples we talked about earlier can be explained by living things coming from other living things. Now, why does Louis Pasteur's name probably look familiar to you? You may have seen his name this morning, in a way, when you were eating breakfast. See, you know why? Yeah, so if you had like cereal and milk, if you look at the milk container, what you'll see is it says that the milk has been pasteurized. Comes from Louis Pasteur's name. You know what does it mean if the milk has been pasteurized? Sean?
Like heated in some way to get rid of the bacteria.
Yes, but we need to go one step further. What else? Ella?
It's heated and then it's like sealed.
Yeah, heated first and then sealed. Heating does what? Kills the bacteria that might be in there, and then sealing it prevents what? What do you need to keep out? Not dust. Correct. Good. So that's what pasteurization is. Heating up a substance. Now it's not just done with milk. Any canned good you buy, you could leave it on your shelf for years, a can of soup, because it's been pasteurized. Before they put it in that can, they heated it up to kill any bacteria inside, sealed it so that no dust can get in there, no new bacteria can get in there, and it will stay fresh for a long time. Okay? Or not fresh, it will stay unspoiled for a long time. So we know today life comes only from other living things. Now, an interesting fact related to this, in early Europe or even early North America, I shouldn't just say Europe, but often water was not safe to drink. Now, sources of water were often contaminated with animal waste or runoff or pollution from settlements and so forth, and it would make people sick. However, when people took that water and turned it into beer, people could drink the beer and not get sick, even though it came from the same water. In fact, they brewed special types of beer that were lower in alcohol content for children. They're called small beers because they provided some nutrition, they provided water without being contaminated and making people sick. So why? So why would the beer made from that water not make people sick, but drinking the water directly would? Abby? Did you ever? Oh, okay. Do you know my answer?
Probably beer was filtered a lot more.
Good idea, but it's not quite because it was filtered. Water.
It was boiled.
Yeah, the process of brewing beer, so I've brewed beer at my house before, is you take a mixture of water, malted barley, a grain, some hops, and you boil it. Put it in a big thing and you boil for a while. And in boiling it, what are you doing to the microbes? Killing them. Then after you brew the beer, you put it in a container and then do what? Seal it up. So really what they were doing was they were pasteurizing the water. Now, let's say they didn't really want to give their children beer that was alcoholic. What could they have done that would have been just as effective? Jake?
Done the same thing they did, but just not add alcohol.
Yeah, or just to use water. Just boil water and seal it. And you would have fresh water that would not make you sick. I mean, if you ever go camping or something and you have to use water from a natural source, one of the things you can do is you can take that water, boil it to kill any bacteria that may be there, and then drink that water. Or if in your neighborhood there's a water main break and there's a possibility your water could have bacteria in it, your neighborhood may be under a boil water advisory, where before you drink any of that water, you're supposed to boil it first. And it's the same reason, same things we just talked about. Any questions about spontaneity?
How long would it take to like, boil the water?
Not that long. How long to kill the bacteria? Basically, it's not so much how long, it's if you bring it up to a boiling temperature, just momentarily, everything that was living in there dies, or most things die. Yeah, and there's different ways to pasteurize. Do you ever see the type of milk that is not refrigerated? It's in like a little box. They sell it in the store, it's just on the shelves. That's milk, it's just like any other milk, but it uses a different form of pasteurization. It's ultra high temperature pasteurized. They heat it up even hotter and immediately seal it, and those boxes of milk can stay fresh on the shelf for weeks and weeks, much longer than milk that's pasteurized by other means.
So it's better?
Different, has a longer shelf life. It does change the flavor somewhat, because those high temperatures change some of the proteins in the milk, so it tastes a little different. A lot of people don't like the taste of it. When I was in Italy last summer, almost all of the milk they sold in Italy was that type of milk, packaged in on the shelf rather than refrigerated. They did have some refrigerated milk, but it was more common to have the on the shelf sort of milk. All right, so let's just finish up today. You finish up by answering those questions on the next 2 slides. Are there 2 slides? Yeah. So answer those questions and we'll talk about them, and also give you back your quiz while you're working.